Hello, welcome to Physiology. Well, sorry for my absence tonight. That's why I make this video. And you are basically tonight we finished the exam one, and you had a break, and then we will now start the nervous system because if we don't start the nervous system, we will not have enough time to finish everything. And so just take notes as you used to be, and while you're watching this recorded lecture. We'll start today our nervous system. So you see the same pattern. Usually when I start a organ system, I'll start with the function of the nervous system. It's like a summary of the function, and then I'll have a brief review of the anatomy. A brief review of the anatomy. So let's take a look. The function. The nervous system is to receive, store, and process information. Receives, store, and process information. The information that our nervous system processes is both external and internal. In other words, our nervous system can detect changes in our body. Well, the easiest one is, let's say if you have fever, then you can feel that your body is heat up, or you have a stomach ulcer, you can feel the pain. But the one that I want to give you example is my favorite topic, learning and memory. Learning and memory require all three: receive, store, and process information. Receive is like, okay, how do you study for the exam? Okay, you receive information. You go to lecture, you take notes, you study. That is what you do. You receive. Store. How do you encode the information into your long-term memory? So some people may just repeatedly study the same thing many times, but some of you may try to come up with something personal to you, to try to relate your personal experience to what you learn. Some of you may remember the stupid jokes that I told in the class. All these can help you with memory. And then process information. Process information is like after you store the info, after you form a long-term memory, how do you retrieve it, use them during the exam, which is what happened just now. And then nervous system can both direct and indirect control of your body. For the indirect control, it means we uses endocrine system. So direct control is like what we learn in anatomy. The motor neuron will connect to your skeletal muscle. Then you have a direct control on your skeletal muscle from your brain. And the indirect control of the nervous system is that we will talk a lot about this when we go to the endocrine system, is that your hypothalamus um, secrete a releasing hormone to pituitary glands, and then pituitary glands will secrete pituitary hormone to another endocrine gland, such as an adrenal gland or thyroid gland. And the adrenal gland or thyroid gland, they will secrete their hormone to our body. So that's why we said that the nervous system, in this case the hypothalamus, has an indirect control, because it has to go through the endocrine system first. And then second function, our nervous system control a higher cognitive function, higher mental function. For example, creativity, reasoning or logic, or in language. So I leave some blank behind because I want you to take a guess which side of the brain mostly take care of it. Mostly. Creativity, left or right? Well, take a guess. Okay, the answer is right side. Reasoning and logic, left or right? Take a guess. Okay, answer is left. Language, left or right? Answer is left side. Okay. Number 3, function number 3. Nervous system control autonomic nervous system, which has something to do with sympathetic and parasympathetic. Parasympathetic. So for the sympathetic nervous system is for fight or flight. For the parasympathetic, it do digestion and resting. So that's the brief overview of the function of the nervous system. Okay, after we finish talking about the general function of the nervous system, we'll start the usual review of the anatomy. First of all, so we'll talk about the cell type. Well, in nervous system, you should remember that we have a neuron and something called neuroglia. Neuroglia has 2 other different names. It can be called glial cell or simply glia. Well, so let's talk about neuron. Neuron is like the the cell that make you think. Okay, it's all the interesting things occur. As we can see from the pictures on the screen here, the neuron looks like this. The one on the left hand side is a cartoon. The one on the right hand side, is the this one. I'm talking about this one. This is the real neuron. This real neuron is called pyramidal cell. Pyramidal cell, you can find pyramidal cell from hippocampus, for example. Hippocampus is a structure in the forebrain. It is located in the temporal lobe. Hippocampus in Latin, it means seahorses. So we have two seahorses, one on the left of the temporal lobe, one on the right in the temporal lobe. Hippocampus is for converting short-term memory into long-term memory. So we can see pyramidal cell in the hippocampus. But I will focus on the cartoon here. The cartoon, you can see that it has dendrite, cell body, aka soma. And then we have the initial segment. Okay, I don't like this. Okay, I don't like it. I don't like it is called initial segment. Okay, it doesn't work. I don't like to call it initial segment. Let's call this axon hillock. Can we call it axon hillock? Okay, axon hillock, and then what else? The collateral. We have the axon, of course. Axon is important. Oops. Axon is important. We'll talk more about it, and then at the end is axon terminals. So you see that axon terminal has a different name actually. You may also call it synaptic bouton. Okay, and then so what I want to ask you, the first question is that let me see. So, how come neuron has so many dendritic branches? How come neuron has so many dendritic branches? So, I want you to think about this question. I'll give you 5 seconds. 5, 4, 3, 2, 1. Okay. Well, if you look at the structure of the neuron, you can see that the structure of the neuron actually looks very similar to a tree. So for the dendrite, they looks like the branches of the trees. The axon looks like the dendrite looks like the branches of the tree. The axon looks like the trunk of the trees, and then the axon terminals and synaptic boutons, they look like the roots of the trees. So another way to ask this question is, how come a tree would have so many branches? Well, this may be easier for you to answer. I'll give you another 5 seconds to think about it. 5, 4, 3, 2, 1. Okay, time's up. So some of you may be able to come up the answer why a tree will have so many branches. Well, because you know that leaves grow on the branches of the tree, so that when we have so many branches, then we can have many leaves growing on all the branches. To what? To maximize capturing of the sunlight. Tree, they don't want to waste any sunlight. That's why they have so many branches and so many leaves to maximize the chances of capturing the most of the sunlight. Similarly, what's the function of the dendrite? How come we have so many dendritic branches? It is to maximize the chances of forming synapse. And synapse is basically connection between a nerve cell, connection between neurons. So to maximize the chances of forming synapses. Synapses, they are the connection between neuron. And why is it so important to form more synapses? The more synapses you can form, the so-called the smarter you will be. In other words, let me give you an example. Learning is tough. As you know that whenever you study for the exam, you need to work very hard in order to memorize the stuff, and in order to use it, you know, to understand it. When we work hard to study, the neuron can actually grow more dendrite. Yes, you can have more dendrite growing in the neuron so that you can form more synapses with different neuron or even with the same neuron. Therefore, the more synapses you form, the stronger the memory you can have for that particular knowledge. Therefore, it makes you so-called smarter. Okay, that's the brief introduction of the neuron. Now we go to neuroglia. Do you remember how many different types of neuroglia we have in our body? Do you remember from your anatomies? So I'll give you 5 seconds to think about this. 5, 4, 3, 2, 1. Okay. So correct answer is that we have 6 types. We have 6 types of neuroglia. So in the CNS, I hope you still remember your anatomy. What is CNS? What is CNS? Okay, CNS is basically brain and spinal cord. Okay, in CNS we have astrocyte. Astrocytes, they are star-shaped as the prefix astro. Astro means star, cyte means cells. They are star-shaped neuroglia. What's the function? Do you remember the function of astrocyte? The function is to form blood-brain barrier. Oops. Blood-brain barrier is basically a special form of the capillary. You can only find this type of structure inside the brain. The capillary inside the brain, other than normal capillary, they only have a simple squamous epithelium. That's it. But in the capillary inside the brain, we have of course the simple squamous epithelium forming the structure of the capillary, and then on the surface of it you have some kind of a basal membrane, and the basal membrane, on top of it you have the astrocyte. Astrocyte, the star shape, the star feet wrap around the basal membrane, so that it makes the capillary in your brain less leaky, so it becomes more selective. A blood-brain barrier helps to protect your brain from exposure to, let's say, a lot of harmful chemical, a lot of harmful drug, because it seals up the capillary so that not everything can go through the blood-brain barrier. And that poses a challenge to pharmaceutical company when they want to make a drug. If they want to get to the brain, they have to make a drug that can go through the blood-brain barrier. We'll talk more about this later in the nervous system. Okay, second type. What is the second type of the neuroglia? Second type is something called ependymal cell. Ependymal cell. What's the function? They produce CSF. I don't know if you remember what CSF is, but CSF is actually the cerebrospinal fluid. So they are the solution that provide nutrient to the brain and spinal cord, but also it helps to protect the brain. It absorbs the shock that goes into your CNS. Number 3, what do we have? We have something called microglia. Microglial cells. What's the function? They are, running out of space, let's write it here. They are phagocyte. They are phagocyte, meaning that they have immune function. They eat up any infection they can find, but not, of course, not all. I mean, it's not bulletproof, the protection, but at least we have some form of protection in the brain. So let's say you have microbes that can go through the blood-brain barrier and goes into from the blood to go into the brain. Then the microglial cell, they can eat them up. But of course, not all the microglial cell cannot kill all the infection, but at least you have some form of protection in your brain. Okay, what about, and then fourth one we call it oligodendrocyte. The prefix oligo, it means a few. Dendro means dendrite. So these cells, they have, well, you may call it a few dendrite, but actually what it means is that these cells, they use their dendrite. These neuroglia use their extension or the podia to wrap around the axon of neuron. So they wrap around a few axons. So one oligodendrocyte can use their podia, use their feet to wrap around the axon of a few neurons. So this is what it means. In other words, oligodendrocyte forms the myelin sheath, on the axon of the neuron in the CNS. Okay, so again, oligodendrocyte use their feet to wrap around the axons of a few neurons. Now, what about PNS? Okay, for PNS, you need to say it slowly, otherwise it will become the structure in the reproductive system that we will cover later in the semester. So, peripheral nervous system, PNS. So, what is it? So, basically, everything that are not brain and spinal cord, we call it PNS. In the PNS, what kind of neuroglia we have? Okay, we have, I continue with the numbering 5. We have something called a satellite cell. What's the function of satellite cell? The function of satellite cell is very similar to oligodendrocyte in that it wraps around soma. So but of course, the soma of the neuron, they are in the PNS, not CNS, okay? Last but not least, the famous one, number 6, it is called a Schwann cell. Schwann cell has a very similar function of oligodendrocyte. They provide myelin sheath to the neuron, provide myelin sheath on the axon of the neuron in the PNS. Okay, but remember, oligodendrocytes use their feet to wrap around the axons of a few neuron, but Schwann cell is different. They use their body, the entire Schwann cell. They use their entire body to wrap around the axon. So we can see that in the next slides how it works. Okay, as you can see here, all these 4 is the top panel. The top panel, all these are the transverse section of an axon and the Schwann cell. So the neuron, this is the neuron, the axon is here. Myelin forming cell is basically the Schwann cell. Okay, you can see that the Schwann cell is basically, it uses the entire cell to wrap around the axon. As it goes many round here, basically all these, they are the cell membrane. All these, they are the cell membrane, meaning the phospholipid bilayer. So the cytoplasm is already squeezed to a very thin layer. So it's mostly the phospholipid bilayer here. So when we say that, okay, the myelin, this is the myelin, basically we are referring to the phospholipid bilayer, the cell membrane of Schwann cell. And of course, Schwann cell is a cell, they have nucleus. And in between the Schwann cell, we have something called knock-off-Ranvier. Some people just say it as knock-off-Ranvier. So we will explain to you the significance of knock-off-Ranvier later in the lecture. But now we just get to get a review about the structure of the Schwann cell. Okay, now let's take a look. We have three types of neuron, three classes of neuron. So basically, it is the afferent, the integrate, and the efferent, or afferent. I usually call it efferent to remind me of the spelling differences. So as you know that afferent, the number one, this is the input. Usually, afferent neuron is attached to a receptor. A receptor can be anything like our eyes. Our eyes is a receptor. The taste bud on our tongue is also a receptor. Of course, most of people, whenever they think about receptor, they will think about the receptor on the skin, okay? Like Pacinian corpuscles or Meissner corpuscles, all these are the touch receptor. After the receptor detects the stimulus, they will send action potential to the afferent neuron. So the afferent neuron can carry or transport the action potential, the sensory action potential from the receptor to the integration center. Usually integrate. I shouldn't call it integrate. It should be interneuron. Sorry about it. It should be called interneuron. Interneuron. Sorry about that. Interneuron. They are usually can be found. We have interneuron almost all over the body, but majority of the interneuron we can find it also in the CNS. CNS, what's the function of the interneuron in the CNS? Is for processing the sensory input and also processing the motor output. Okay. And that's why you will have a lot of interneuron inside our brain for us to think about it, okay? The last one is the, efferent. Efferent, okay? Efferent, they are the output. Output usually a motor. It's like a command sent from your brain in response to the stimulus. What we need to do, what we need to execute. So that these three classes of neuron form something called we learned about this, and I test you about this just now from exam 1. Reflex arc. The question is, what are the output of the efferent? What are the output of efferent or afferent neuron? So I may have talked about this when we talk about the reflex arc last few weeks. So let me give a tips. We have 3 types. We have 3 types of output. I'll give you 5 seconds to think about this. 5, 4, 3, 2, 1. Okay, time's up. Output of the efferent neuron. It can be another neuron. It can be. There you go. It can be a gland, or it can be a muscle. So these are the 3 target of a neuron. So as you can see on the screen here, we have three classes of neurons. Okay, you have the afferent, you have the interneuron, and you have the afferent, or I personally call it efferent. Okay. So as I mentioned before, afferent neuron usually connects the receptor to the CNS. Okay. That means your brain and spinal cord. And yes, the afferent neuron and the efferent or afferent neuron, afferent and efferent neuron, they are all in the PNS. So receptor is also in the PNS. And then efferent neuron carries the axon terminal here connects to the neuron, glands, and muscle. And then as I mentioned, this is the sensory input, and this is the usually the motor output. And we do all the processing or thinking in here. And take a guess, what's the ratio between the 3? So the ratio between the 3, afferent, efferent versus inter. Take a guess, which type of neuron is the most, which type is the least in terms of number in our body. In our body we have the most, which one? Okay, I'll give you 5 seconds to think about this. 5, 4, 3, 2, 1. Okay, time's up. Most of you hopefully will get it correct. We have the most is the interneuron. The least we have is actually the afferent neuron. The ratio is approximately for one afferent neuron, we have approximately maybe 10 efferent neuron. And for interneuron, we have approximately, if it is in this ratio, at least 200,000, at least. And then and remember we mentioned about the, what do we call this part here? Or what do we call this part here? Do you remember the name of this part? We briefly mentioned about this in previous slides. I'll let you think about this. What's the name of these two parts? They are called synapse. Definition of synapse is a connection between two neurons. And then how do they communicate? How do they communicate? For this type of synapse, we have two types of synapses. One is called electrical, one is called chemical. In this part of the course, we will focus on the chemical synapse. The chemical synapse uses something called a neurotransmitter. So from now on, I'll just call it NT. Neurotransmitter, they're chemical that is released at the axon terminal. This is also an axon terminal. In other words, this is also a chemical synapse here. An axon terminal, and then it will bind to the receptor on the soma of another cell. Soma, or the dendrite. Theoretically, it should be the dendrite of the other cell. So, what's the definition of neurotransmitter? Yeah, it's a chemical messenger used by neurons to communicate with each other or effectors. Again, effectors, you know that effectors, they are glands and muscles. Okay, starting from here, we'll go to the real physiology. Okay, for all the previous things that we talked about, they are the review of anatomy. Now we start the real physiology. The first thing we need to talk about is the concept. It's called membrane potential. The first thing we need to talk about membrane potential is something called resting membrane potential. We will call it RMP from now on. Resting membrane potential, what does it mean? Well, to put it in a very simple way, when a neuron is not doing anything, then the neuron is at resting membrane potential. When the neuron is not doing anything, the neuron is at resting membrane potential. What is the value of resting membrane potential in human neuron? It is approximately -70 millivolt. Okay, well, let's do it again. Sorry. So for the things I want you to memorize, I usually do it in blue. You'll see that for the rest of the lecture, for the rest of the semester, for the things I want you to memorize, I'll do it in blue, and then I'll put a box around it, and to make it even more conspicuous, I'll put stars here. Yes, I want you to memorize this. In the later of the lecture, when we talk about cardiovascular system, respiratory system, you have a lot of equation you need to memorize. Then I also do the same thing, blue with a box and then with stars next to it. So yes, you need to memorize that. And how do scientists find out that when a neuron is not doing anything, the resting membrane potential is minus 70 millivolt? Well, they use something called a voltmeter. Voltmeter is basically a machine that can measure the voltage of the neuron. A voltage is, you may call it a driving force of a current. So you may have heard about voltage and current. Current is basically moving of the electron, we call it current. But the driving force, or how fast the electron moves, is depends on the voltage. So this voltage is a driving force for the current. On the voltmeter, you have two electrodes. One is called intracellular, one is called extracellular. But in a research setting, we call it electrophysiology that we usually do not call it sometimes we call it intracellular electrode, but most commonly we call this the recording electrode, and we call this extracellular one, we call it grounding or reference electrode. It's either grounding electrode or reference electrode. So what does it do? The grounding or reference electrode, it always, when we put it in the extracellular fluid here, when we put the grounding electrode in extracellular fluid here, basically I should say that whatever the grounding or the reference electrode is, they always assume that the value is equal to 0 millivolt. That's how we call grounding. Ground is like, okay, we assume that this medium, in this case is called ECF, extracellular fluid, they assume that ECF is 0. How do they assume that? Oh, by putting the grounding electrode or reference electrode in the ECF. So that we use the ECF as the reference. That's how we call grounding electrode, reference electrode. Whatever this electrode put into, we use that as a reference. How do we use that as a reference? We assume that it is 0 millivolt. And then the recording electrode, whatever the recording electrode is in, then we compare the value the recording electrode get with the grounding or reference electrode. So basically the value that is shown on the voltmeter is the relative comparison between the recording electrode with the grounding electrode. And you can see that in the cell, you have all these negative charges here, and the recording electrode is inside the cell. So the recording electrode can measure the negative charges inside the cell. That's why it is negative here. And the voltage difference is that the inside the cell it is a 70 millivolt. Okay, in compared to the ECF, in compared to the medium the grounding electrode is in. So that's how scientists get the resting membrane potential as minus 70 millivolt. So let's see. Okay. Before we take a break, this is the last one. It's like a review, and I also want to give you a brief so-called review of the physics. So the first review is about what we talked last week when we talk about how gamma ray works, when we talk about the primary active transport and the secondary active transport. Remember I told you to memorize the concentration of the sodium and potassium. So it is in here. The concentration of sodium is high extracellularly but low intracellularly. And I gave you the evolutionary explanation for that. And then the potassium is the opposite. Outside of the cell, we have low potassium concentration. Inside of the cell, we have high potassium concentration. Now, I want you to memorize one more ion. It is the chloride ion. Inside of the cell, we have very high concentration of chloride, and outside of the cell, we have low concentration inside of the cell. So, how do you memorize chloride? Well, use the same evolutionary explanation I told you last week. Remember that we all came from the ocean, and then when we migrate from the ocean to the land, we have to enclose some of the seawater with us so that we won't get desiccated. Sodium, of course, comes from our ocean, because our ocean has a lot of sodium chloride. Yes, because we have lots of sodium in the ocean, we also have a lot of chloride ion in the ocean. So when we enclose the seawater from the ocean, then our extracellular fluid has a lot of sodium ion and chloride ion. So that's how you memorize it. Now, the review of the physics. I don't know how many of you have taken physics, like physics 105, physics 106 in Glendale here. The reason why it is helpful to take some physics, because you need to know the relationship of voltage and current. Okay, current. We use the symbol I for current. Current is basically movement of charged particle. So what are the charged particles? So in everyday life, you have battery, okay? AA battery, AAA battery, or 9-volt battery. So what happens is that the battery, they will move what kind of charged particle? The battery will move the electron. Okay, the movement of electron is like the current. What about in the nervous system, our neuron? In the neuron, the movement of the charged particle, we call that ions. Okay, in the neuron, we will focus on two ions. Sodium and potassium. So the movement of the ion, sodium ion and potassium ion, we will have the current. Okay, sodium current, potassium current. And then another important concept, basic concept that you need to learn from your physics is voltage. We use the symbol V. Voltage, you might call it the driving force of current. Voltage is the driving force of current. What do I mean by driving force? You have the movement of the charged particle, movement of the ions, movement of the sodium ion, let's say. But how fast the ion move? How fast the sodium ion moves depends on the voltage. What is voltage in this case? The voltage is actually the differences of the concentration between extracellular fluid and intracellular fluid. This is the voltage. Voltage of the sodium, for the sodium ion. So the voltage is the differences of the concentration between the extracellular fluid and intracellular fluid. In other words, different ions, they have different voltage. Depends on the concentration of the ion. For example, let me ask you this question, see if you understand what I'm talking about. For chloride ion and potassium ion, which one has a higher voltage? Which one has a lower voltage? see if you can answer this question correctly. Which one has a higher voltage? Which one has a lower voltage? I'll give you 5 seconds to think about it. You can discuss it with your neighbor. Okay, I'll give you 10 seconds. You can 5 seconds to think about your answer. The other 5 seconds you discuss it with your neighbor. All right, 5, 4, 3, 2, 1. Okay, start discussion. 5, 4, 3, 2, 1. Okay, time's up. We probably don't have enough time to discuss anyway. Okay, so which one has a higher voltage? What's the differences between the potassium here? The differences here is like 144, 145. The differences here is 100. So in other words, the voltage of the potassium ion is larger than the voltage of the chloride ion. Does it make sense? In other words, potassium ion can move across the membrane faster than chloride ion can move across the membrane because potassium has a stronger or higher driving force. So that's the basic concept between voltage and current that you will repeatedly see it in our discussion, in the lecture, in the next few lecture. So let's take a break. Let's take 5 minutes break from here before we start something very important. We call it resting membrane potential.